So today I'm going to talk to you about a little bit of work I did with my PhD dissertation, and specifically I kind of piggybacked on some of the work that Dr. Bill Schillinger and Dr. Tim Politz had done. They had a long-term trial set up in Davenport, Washington, where they were monitoring the yields of spring wheat following canola. And so that's kind of the basis of the work that I'll be reporting on today. I'd like to acknowledge the contributors to this work. The members of my committee were Dr. Bill Schillinger, Dr. Tara Sullivan, and Dr. Tim Politz. Alrighty, so a lot of this material on this slide was actually already covered by Lynn Carpenter-Boggs, but essentially what I'm talking about is plant microbe interactions, and those can either be positive, null, or negative. And I'm going to report on some of the things that we saw in the microbial community following a canola crop. And to get me going, if you've seen any of my talks, I like to include a couple of jokes just to get the nerves out. So we'll start with this one. Why don't plants like math? Well, because it gives them square roots. I tried these with my kids last night. They kind of laughed. Well, how about this one? So as a scientist, what do you do when you tell a joke and nobody responds? You keep going until you get a reaction, right? And last but not least, we had a graduate student this semester who spent a lot of time looking in the microscope, and so I'm sure she saw some of those anaerobic bacteria that weren't doing much, but the aerobic bacteria were doing leg lifts and jumping jacks and other stuff that was more aerobic. So with that, the first disclaimer I'll make is the work that we were doing was looking at canola as a rotational crop. A lot of times when you hear about the biofumigation potential of canola and other brassicas, sometimes that work is referring to canola as a canola or mustard as grown as a green manure that is then chopped up, macerated, cells are ruptured and incorporated into the soil. That gives a maximum biofumigation effect. The research that we were doing was just looking at the presence of the plant grown to maturity that was harvested, and what is the effect of that plant in rotation with cereal grains. We've heard a lot about the active effect of a brassica grown in rotation. Some of those are that you'll see, well, first of all, we hear a lot about glucosinolates, and as they exude from roots or from residues of canola, they'll form a compound called isothiocyanase, and that's what is responsible for the biofumigation effect. A lot of other things that are accompanied with the presence of a canola plant, and in addition to glucosinolates, there are other compounds. One of those is referred to as dimethyl disulfide, which also has a biofumigation effect. And so the potential exists for these compounds to have an impact on the soil microbial community, particularly because when you look at the concentration of glucosinolates within a canola plant, the concentration of glucosinolates is actually higher in the root system. And those glucosinolates are leached out as the plant grows. And so that root system is in direct contact with soil microbes. And if one of the claims to growing canola in rotation is to set back pathogens, either fungal or bacterial, then those compounds aren't necessarily selective, and so they're obviously going to have an effect on beneficials that they come in contact with as well. We've all heard about a lot of the rotational effects of canola, and one of those is you expect to see a yield increase in a grain crop following canola. That can be attributed to a number of things, but one of those is setting back pathogens. You might have a change in nutrient supply following canola with higher levels of nitrogen following canola. I won't spend a lot of time on the rotational benefits because we've probably all heard about those. But what we hear less about are the reported negative effects of canola on either yield or microbes that they're in contact with. And as I mentioned, I did work with Bill Schillinger and Tim Politz, and essentially while they were conducting their work where they were looking at a yield trial of spring wheat following canola, I was collecting soil samples at the same time, which was very fortunate because we would then be able to go back and look at what was happening with the microbial community with canola in rotation. So that kind of leads to the justification for this research. A lot of growers were curious about what was happening to soil health when you include canola in rotation. And then also the work that Tim Politz and Bill Schillinger done, they reported over a 7-year period. And this is, first I'll say where the study was conducted. It was up in Davenport. It's in the intermediate rainfall zone in an area where the grower was trying to go to an annual cropping system where they were having, I guess, 2 crops out of 3 years as opposed to a fallow cropping. They would have a fallow period 1 and 3 years. And the rotation was winter wheat, spring wheat, and no-till fallow compared to winter canola, spring wheat, and no-till fallow. And so they were following canola. They were following spring wheat directly after canola with no fallow period. And over that 7-year period, they observed a reduction in spring wheat yield of about 17% following canola. And so the work that they did ruled out moisture, ruled out plant pathogens, ruled out nutrient supply. And so that kind of led us back to there's got to be something going on with the microbial community leading to this yield lag. So from that, that first study up there is the one that I've just referenced. And we also established a study in the Ritzville and Mansfield area where we went on growers' fields and we identified growers that were growing canola and wheat next to each other, where we could go in and sample and look at particularly in the Rhizosphere and what's going on with the microbial community. And then we also there'd been a lot of talk about Camelina and the effect of Camelina, and Bill Schillinger had a trial at the Lind field station where we conducted similar work. We went in and looked at how things were responding with with Camelina in rotation. And so I'll just kind of discuss the results of those 3 studies today. First, the analysis that we were looking at, Dr. Huggins referred to a little bit ago, is I was looking at the phospholipid fatty acid profiles, and what those are is every organism that has a cell wall, that cell wall contains a phospholipid bilayer, and those are carbon chains that are characteristic to certain microbes that are in the soil. And so we can start to identify different microbial groups and their changes in quantity and how the community shifts just looking at those lipids that we extract. We also did do some sequencing work in Dr. Sullivan's lab and Dr. Pollitt's lab, and then we did some enzyme assay work. So I threw this graph up just because this means absolutely nothing to you. These are chromatograms of the lipids that we would get off of a machine. But what I want to relate this to, these are sometimes referred to as a soil fingerprint. What I want to relate this to is maybe a human population. So when you think about microbial populations, they're a lot like human populations. When you talk about Pullman, Washington, and you compare that to Seattle, Washington, there's obvious differences in that population. There's obvious differences in abundance. There's a lot more people that live in Seattle. But the makeup of the community in Pullman and Moscow is probably a little bit different, just because of the nature of the industry that's here. We do have 2 universities within 8 miles of each other. That's gonna foster a lot of people that have higher education, as opposed to maybe other communities. And so when we look at these phospholipid fatty acids, we're just trying to get an idea of 1, how many microbes exist in the soil, and 2, what is the composition of that community, and how are the community members contributing to each other. This graph, you can't really see it, but this would be Seattle, and these green dots are associated with the level of education. This is Moscow Pullman, so you can see the population is a lot sparser, but there is, what you can't see is a higher population of college level graduates. Sorry, going back here, the higher level of education in Seattle is concentrated around the downtown area, and then you get out in the outlying areas and you see education levels dropping. And so I throw this up just as a demonstration of what the kind of information we're trying to gather when we look at soil microbial communities. So the first experiment, which I've referred to, is essentially it was winter wheat and winter canola grown side by side in the first year, and then spring wheat was grown directly over both of those treatments. And the yields of those, plus a lot of other data was collected. What I'm going to report here is just the abundance data, how many microbes were there, and what happened to their populations after being exposed to canola and rotation. And so we have these first two bars here. So these are the microbial groups that I'm looking at. So we got fungi, AM fungi, which we heard a lot about this morning, and then we've got gram-positive and gram-negative bacteria. And then this last indicator here is total lipids, and so that's just a summation of everything. And when we look at total PLFA, that's a lot of time referred to as what we would call the viable microbial biomass. And so from here on out, I'll try to refer to them as microbial biomass, because it's a little bit more relatable. But so in these first two bars would represent the winter wheat and winter canola in the first year of the study. And you can see with the side by side, we see a reduction in the fungi as well as the AM fungi in winter canola compared to winter wheat. These next two bars represent the second year where we are looking at spring wheat following either winter canola or winter wheat. And that residual effect carries over into the second year, or at least the second crop year, where we see a reduction in fungi and AM fungi. The same trend carries across bacteria as well as total, but in most cases, some cases, they aren't always significant. But it was interesting to see this trend of reduced microbial biomass in canola following, in crops following canola, as well as in the active canola crop. These are just graphs that now moving from abundance, well, what about the community and the makeup of that community? Are those communities different? And yes, indeed, they are. And so these are the communities that are associated with winter wheat. So we've got winter wheat, and then we've got spring wheat following winter wheat. These are the communities of winter canola and spring wheat following winter canola. These circles represent where these circles intersect. That would say that those communities aren't significantly different, but where they do not intersect, we can say that they are different. And so you can see the separation between the treatments that are involved in winter canola. These arrows here give us an idea of, well, who's responsible for that separation or that change in the community. And almost across the board, total PLFA, just total microbial biomass, is always a big indicator. But just like the previous slide, we see large contribution from the fungi and AM fungi. And that actually, to me, makes a lot of sense, because what we hear a lot about with brassica crops is the suppression of fungal pathogens. Well, like I said earlier, if you're going to suppress a fungal pathogen, you might suppress beneficial members of the fungal community as well. So moving on to, well, that previous study was collected from bulk soil. So we weren't targeting the rhizosphere. So we did establish a study where we were looking at rhizosphere soil. And to further distinguish on Lynn's definition of the rhizosphere, so I'm looking at the soil directly around the root and what adheres to the root, but then there's also rhizoplane and other definitions, and we have ongoing work where Dr. Tim Paulitz is actually looking at rhizoplane bacteria and fungi. So just further distinctions, this is rhizosphere, so this is a soil adhering to the root itself. We saw similar trends, and just a little bit of background on this study. So we went out into established crops in the fall. So it was about mid-November. Plants had established. We collected rhizosphere soil, and you don't see a lot of differences in the communities in the fall. So everybody's just kind of how they were at seeding. We came back in the spring after plants had an opportunity to develop a little bit more, and we start to see similar shifts in the community where we see reductions in fungi and AM fungi, as well as bacteria and total PLFA, which is kind of what we expected. We saw that in bulk soil. I expected to see it a little bit more pronounced in the rhizosphere, but we did move from Davenport out to Ritzville and up in Mansfield, where we have a little bit sandier soils. So, but overall, the trend of a reduced microbial community. Again, in the fall, this correlates with the previous slide where we don't see a lot of separation in the communities. The separation that we do see is associated with the landscape. We've collected from the ridgetop and the bottomland. But when we move into the spring, we start to see separation that is associated with crops. So we've got our wheat here and our canola here. And so with further plant development, as these roots exude compounds, you start to see shifts in the microbial community and reductions in the microbial biomass. Just summary from the DNA sequencing work from the same study, Rhizosphere soil, same kind of thing. We did see shifts in bacterial taxica. Those bacteria could potentially be plant beneficial. So I'll refer back to Dr. Boggs' analogy of the silver bullet, and Tim Paul has said this before. We don't have a smoking gun or a silver bullet, but there are a lot of things. The silver buckshot, right? So this might be a buckshot effect where there's other things contributing to this. We didn't pin it down to one thing, but there's certainly something going on in the microbial community. Moving into the Camelina study, which was interesting. This was out at Lind, and so more of a fallow type cropping system. And the traditional rotation is represented here. These two bars are winter wheat and summer fallow. So that's the rotation that's been practiced since the induction of agriculture in that area. And comparing that to the rotation of winter wheat, Camelina, and summer fallow. And so Camelina, similar to canola, and in some cases might even be a little bit hotter as far as glucosinolate content. What we see in the microbial community is this stepwise reduction. We see a reduction going from winter wheat to Camelina, and then a further reduction going from Camelina to summer fallow. So what I found interesting was that this Camelina, this reduction in Camelina was similar to what we saw in the reduction going from winter wheat to summer fallow. But it was also interesting to see this additional reduction with, so it's, I guess I refer to it kind of as a 1-2 punch, where you're throwing in a different crop that has these compounds associated with, and then you're following that with summer fallow. And so you're, one, have a non-host, you have a non-host that has glucosinolates associated with it, but then you're following that with summer fallow where you don't have the actual presence of a plant, and so you're going to see a further decline in the microbial community. And that was kind of consistent across the board. But in all cases, including the canola data that I previously looked at, we saw a rebound in the third year. And so this wasn't something that led to an internal decline in the microbial community. There's a reference to if you build it, they'll come back, or they will come. And so the soils are very resilient. And in the case of the Davenport study, in the third year, we did not see a reduction in yield, nor did we see a reduction in the microbial community. And so it was kind of a short-term effect where we saw this reduction, saw a slight reduction in yield, but then everything kind of rebounded. And so in that case, speaking of inoculants, you know, in that third year, it wouldn't benefit you to try to resolve it because it's probably going to resolve itself. And I compare that to this little picture I have here. So when we go to the mall, my kids try to go up the escalator, but they try to go the opposite way, and they don't ever make any ground. They take a step down, but they always end up at the same point because the staircase is moving up. And I draw an analogy of that to the microbial community. You might, if you have a resilient soil where you have a healthy microbial community, you might take a step down, you might get a short-term setback, but if through sustainable practices, you're going to end up back in the same spot. And some of the research I've looked at in Canada, where you have continuous canola cropping, then you start to see this decline that precedes, and you start to foster a different microbial community. You start to foster pathogens that are associated with canola. So I didn't want to be too alarming with the data that I presented. We did see a reduction in the microbial community, but it seemed to recover. Just some of the work that we're conducting now, and I'll give a shout out to Dr. Dave Huggins' graduate student, Cassandra Reeser. She has done a lot of work in this past year out in Ritzville, where Dr. Schillinger has observed some yield decline following canola, and she established a set of subplots where we were looking at the what was going on with spring wheat following a number of different crops, following spring barley, continuous spring wheat, triticale, canola, and winter wheat. And this is preliminary data. We were, what we did is we went in after these crops and we marked out a subplot of that area, and we did hand inoculations with AMF inoculum. And this was very painstaking. We were injecting AMF below the seed that had been seeded and by hand with these big hunkin' syringes, and then going out as plants develop and collecting plant samples as well as soil samples to see if we could get an idea of whether an inoculation would resolve some of these concerns. Across the board, you can see that there was a response, a yield response to the inoculation. Those were not significant necessarily. Here it was significant following triticale. What we expected to see here, at least I was hoping to see, is that we would see a response to inoculating following canola, but we saw the opposite. At least, well, these are not statistically different, but the trend was that the inoculated side did not yield better than the control side in spring wheat following canola. So this might go back to a lot of the things that Dr. Boggs was talking about, where it maybe is our inoculum, maybe it's our timing of application. We are going to continue this work because we think it's worth pursuing to just try to solve the question of if there is a temporary yield lag, one, how do we identify where that's going to happen, and how can we correct that just so that we can keep things profitable? I'm getting short on time, so let's see, I guess takeaway messages are that you know, this yield reduction was short-lived. The setback in the microbial community was short-lived. That might not be the case if you have a soil that's not as resilient. You might see a more long-term effect, but as long as we continue with our sustainable practices of trying to foster soil systems that have, trying to increase organic matter and trying to feed the microbes that we have within our soil system, any setback that we see, we're going to probably recover from, and it's not a big concern. So, I don't even know what's next. Oh yeah, so just acknowledge all the people that I've worked with. Obviously didn't do this on my own, but grateful for Bill Schillinger, Tim Politz, and their work, and Bill Schillinger's crew, which maintained the plots, as well as the graduate students that I've been working with here, and all the advice that I've received. And with that, I'll take any questions if we have time for questions. Yes?
Do you anticipate seeing the same thing? All this was with winter canola. Do you anticipate seeing the same thing with spring canola?
Good question. So the question was, we saw this with winter canola. Would we anticipate seeing the same thing with spring canola? And Dr. Schillinger actually has plots in Ritzville where they've planted winter canola, and in 2017 and 2018 the winter canola failed, and so they ended up planting spring canola. And so I guess not necessarily planned, but they did have a spring canola treatment, and in 2017 and 2018 they did see a reduction in yield in spring wheat following those. But associated with that data, they also did see differences in moisture content. And so, so when you see differences in moisture, you can point the finger at that. So this might be one of those combination effects of moisture and microbial community. In 2019, it was spring canola. They didn't see a reduction in yield.